The cell tracking wizard is used to analyze samples stained with CFSE or other cell tracking dyes. In this tutorial, we'll create a cell tracking project to demonstrate how it works. I have ModFit LT running here, and we'll start by creating a new project. Click the New button in the project group. The type of project that we're going to create is a cell proliferation tracking project. We'll give it a name. The project will be located in the projects folder, which is in the ModFit LT folder of your documents location. And I'm going to open the More group here and enable the Copy Data Files option. This will copy any files that I add into my project folder, so I have them locally. We click OK. And our project is initially set up. We have a blank report page. We have a place where we will eventually see the files that we're adding to be analyzed. And we have a group of command states here which will tell us what to do when we analyze each file. Our next step is to select the files to analyze. We use OpenFCS to do that. I click the button. These files are located in the samples proliferation folder within the location where ModFit LT was installed on your computer. And I'm going to select these two. When I select those, they are added to our batch list over here, and the program reads the first file. It's now asking us in this analysis setup to select which measurement we want to analyze. If we want to use FL2 height. This is our cell tracking measurement. We don't need a gate here. If we did, we could use the Add Gate button to do that. I'm going to click OK. And the file completes reading. We see our ungated cell tracking measurement on the report. Next, our attention switches to selecting what operations we want to perform when we analyze each file. So that's done in this panel. Here again is our cell proliferation tracking type of project. And the type of analysis it's going to do, it will fit with the current model, which will be a model created by the cell tracking wizard. We want to save a report for each sample that we analyze. We'll also save a PDF. Those will be saved into the project output folder, and you have other options that you can choose to save here as well and to print. At this point, we're ready to set up our cell tracking model using the Cell Track Wizard. I'll click that button. The start page, we can simply start this model from scratch, and we move to the parent page. On this page, the program is looking for the brightest peak in the sample. In this case, there's only one peak. And it will place this red cursor over the peak that it finds. If it doesn't find the right peak, you can drag the cursor to a different location. You can also type in the position here. And that's all we need to do on this tab right now. In other experiments that you do, you may need to enable the Lock SD option with noisy data. Let's move to the Generations tab. Here's where we tell the program how many daughter generations we are hoping to analyze and find. It's set for 9 by default, and as you can see with these gray dotted lines representing the locations for those daughter generations, 9 is the maximum that we can get in this dynamic range that we've got with this sample. The model option is set for standard. The other option is floating, which is allowing more flexibility in where these generations fall. We'll leave it on the standard model for now. Moving to the other tab, this is where we can choose the generation that defines the precursor frequency and the upper generation PI statistic. And this is set by default to the published standard of generation 3. There's an option for modeling background, which is done with a rectangle stretching across the full range of the model. And if that's enabled, there's an option to enable asymmetric cytokinesis, which interpolates the standard deviation of each generation. We can export a partition table. If that's enabled, we can enable the prompt for file name. We'll click the Analyze button. At this point, the wizard creates the model based on those decisions that we made. We can see a single peak with a little shoulder population here. There's a legend telling us what each color means, and currently the majority of it is colored this dark blue for the parent. We have some demographic information, information about the parent's fraction and position, and then each of the daughter generations listed here. Here are the key statistics that you are expecting to find when you're working with cell tracking analysis. It's a good time to make any adjustments to your report. For example, we could select this image and make it a little bit bigger, just for visualization. If we do make any changes, it's a good idea to save your project so that those changes are propagated into other analyses that you do. We get a prompt here. Do we want to save an updated report template? That will store this position, any changes that we've made here. Now, before we move on to analyzing our next file, let's create a database so that we can store these results for each of the files that we analyze. To do that, we'll click on the Edit tab and enable the Database Wizard. On the Database Wizard, we have a Welcome tab that tells us what we can do. We're going to click Next. We're going to select the database that was created for this project and click Finish. When we do that, the Database Wizard adds 70 fields to our database, and it displays a viewer to make it convenient for us to see what's in there. We are now ready to analyze our next file, and it is at that point where we'll see the database add a record for the current file. To do that, we'll click the Next button on the batch toolbar. Next file is read and analyzed, so in this case now we can see most of the cells are not in the parent anymore. They have divided one time or more than one time into different daughter generations. Our analysis results reflect those visual differences with fractions that are now not zero for many of the generations. And our database has two records. It added the one for the first file, and because this is the last file, it automatically added a record for that as well. So we've actually analyzed all the files in our batch. How would you go back and look at the analysis for one of the previous files if you wanted to review that? The easiest way to do that is to double-click on the file in the list. Let's try that with this first file. And here we're given a choice of opening a PDF or opening the report that ModFit LT did, or we can reanalyze the file if we want to. The PDF will be displayed in your default PDF viewing application, and the report will reload the finished analysis of that file. If you get to a stage where you need to make modifications to your cell tracking model, use the Cell Track Wizard button on the Wizard toolbar to redisplay it. You can edit the model or start from scratch, and visit each tab and make any changes that you need to make. For example, we could switch this to the floating model. When we're done making our changes, we can choose Analyze. This again would be a good time to save any changes that we've made, and you can then reprocess files in your batch. So that's an introduction to using cell tracking wizard projects in ModFit LT.